In prokaryotic cells, translation happens simultaneously with transcription. For this example, though, we will be talking about translation in eukaryotic cells. Starting off, mRNA will leave the nucleus through nuclear pores and then enter on into the cytoplasm. A ribosome will then attach to the mRNA, and the ribosome is also responsible for translating the nucleotide sequence of an mRNA molecule into the correct amino acid sequence for a polypeptide. This is done through reading the codons on mRNA, a 3-nucleotide sequence that codes for specific amino acids based on their order and types of nucleotides. tRNA has the nucleotide triplet to match the mRNA, known as an anticodon, which allows specific and proper amino acids to be brought into the polypeptide chain later on. An enzyme called aminoacyl tRNA will attach the amino acid to the tRNA, leaving us with a tRNA ready for protein synthesis. Moving on to the ribosome, ribosomes are made from about 1/3 proteins and 2/3 ribosomal RNA. The ribosome also has many other components, having a large subunit containing an A, P, and E site with an exit tunnel, a small subunit, and mRNA binding site. The aminoacyl tRNA binding site, or A site, holds the tRNA in place while waiting for the amino acid to be added to the polypeptide chain. The peptidyl tRNA binding site, or P site, holds the tRNA that's attached to the growing polypeptide chain that will leave through the exit tunnel. Finally, we have the E site, or exit site, where the tRNA leaves the ribosome. Starting off, the small ribosomal subunit binds to mRNA that travels from 5' to 3'. An initiator tRNA will then bond to the start codon, causing the large ribosomal subunit to attach, completing the translation initiation complex. The complex does its job, properly reorganizing codons, forming peptide bonds in the polypeptide chain, and sending tRNA off to get another amino acid. Polyribosomes, or a chain of multiple ribosomes, are able to read the same strand of mRNA to produce multiple of the same proteins at the same time.